Thomas enjoys trapping Jenny wrens in corrugated iron and then molesting them with coriander stems. Recently, he has, unbeknownst to his co-workers, spiked their breakfast cereal with the eggs of some parasitic tropical worms, which they have now ingested, and which in time will make their way to their brains and bore deep inside them. Mr. Lars Thomas and his parasitic brain worm trickery.
I suppose you've already noticed how my camera crew has been behaving very strangely in the last couple of days. Well, now I'm going to tell you something very serious and very scientific. No, actually, I'm going to tell you about how I do those hair analyses that I have been going on about for the last couple of days. As you can see, I am calling my lecture A Hair of the Beast. But as you can see further down, if you read further down, I don't know if you can actually read that. But when you do hair analysis, you have this strange tendency to collect hair samples wherever you go, because you need to have something to, to compare your unknown hair samples with. And people tend to look at you very strangely when they say, oh, by the way, did you see my son has a new rabbit? And I say, oh yes, thank you. Can I see this rabbit? And as soon as I see this rabbit, oh, it's very nice. Can I yank out a tuft of its hair? And usually the kids are terrified, but in that way I have amassed a vast collection of hair samples from all kinds of things. Well, to make everything sound even more scientific, the study of hairs actually has it's a scientific discipline all of its own, called trichology. When you study hair or trichology, you try to, for instance, determine the structure of hairs, how you can identify them, the use of hair, and various other things, like how disease will make hairs look different or develop in a different way. There's all kinds of things that you can do. Hairs, we mammals have hairs, and it's mammalian hair that I'm going to talk about. Carl Portman will tell you that spiders have hairs as well, and you can study the structure of those. But here today, it's all about mammalian hairs. The hair that we have and all our fellow mammals have several different functions. They can be used for insulation. You see this polar bear, and they have this magnificent fur that makes it possible for them to live in the Arctic. I must confess, though, that polar bear fur, when it's sitting on the outside of a live polar bear, can have the extreme opposite effect, because when you're sitting in a bus and suddenly you see this outside your window, you tend to feel very, very cold very, very quickly. Another function of the mammalian hair is a sensory organ. This is Boris, and he belongs to a friend of mine. And I'm not sure if you can actually see his eyebrows and his guard hairs and so on, but they are a sensory organ in this case. And they are also hell to identify if you find them. They can also be used as the color of the hairs are camouflage. This is an African antelope, and although it's extremely visible in this picture, when they are in the dense forest where they live normally, it's very, very difficult to see them. This one, luckily, was used to people, so it came out of the forest. Every now and then it went into the back garden of a friend of mine who lives in Africa, and so I was able to take this picture of this creature. And hairs, of course, can have a signaling function. Various animals have, tufts and combs and stuff, they can raise their hairs. Anyone who has a cat know how a cat makes their hair stand on end when they want to look big and fierce and more dangerous. Or a few mammals. Mammals are usually rather dull in color, but a few mammals are extremely colorful, like this golden line tamarin that you see in this image. But I am going to talk about how to identify hair samples. It sounds like a rather esoteric subject, but for cryptozoologists it's of the utmost importance, because very often the only evidence we have for various creatures is a few tufts of hair found on a branch, on a bramble somewhere. And I know of a lot of cryptozoologists who have lots and lots and lots of hair samples and would very much like to identify them. And it is quite possible to do so with quite a lot of precision. A hair, to the left you see an electron microscope image of the outside of a hair. It looks more or less like a tree trunk with the bark falling off. That's a very thin layer of scales made of protein. And the inside of the hair, you will find the medulla in the center, the central part of the hair. That's usually where you find the pigmentation. And then there's an outer layer, and there's this layer of scales on the outside. By examining these various features in detail, you can actually identify most mammals. It takes a bit of practice, but it is fairly easy when you've done it a few times. And I think it's important that, well, I think it should be a compulsory course for every cryptozoologist, hair identification 1. Because today the kind of holy grail, or if you like, of any identification is DNA analysis, symbolized by this drawing of a DNA molecule. DNA analysis is of course very precise, but it's not as expensive as it used to be, but it is fairly expensive to have a DNA analysis done. So if you're an amateur cryptozoologist with 250 hair samples in your collection, it can be a bit overwhelming to even imagine how to get more identified. But there was a time, strangely enough, where DNA analysis was unknown, and you would still be able to identify animals, identify hairs. And of course you can still do that. So I am going to give you a crash course in how to identify hair and give you an idea of what I have been doing for the last couple of days when I come down here and tell you that I found dog, a woman's hair, a badger hair, and indeed leopard hairs. Just to give you an idea of the variation that you can actually find in a single hair. Well, I can hardly see what's up there, so I don't know if any of you actually can, but this is the surface pattern, various surface patterns on hairs. These protein scales that I told you about can, on a fresh hair, have all kinds of patterns like bricks or on an old wall, or look like the side of a palm tree, or bark falling off an old oak tree, or whatever. And they can also give you an idea of how old the hairs you have found actually are. Because in a few days' time, sometimes only in a few hours' time, they start to fall off. And if you find a very old hair, all these scales will be gone. For instance, those leopard hairs that we found in the forest a couple of days ago, and which I identified yesterday, had been up there for some time, because a lot of these protein scales had fallen off. So it wasn't just the day before yesterday this leopard had gone past this bramble where we found the hairs. And another thing you can look at is the structure of the medulla, which is the central part of the hair, where you see the various typical, where most of the pigmentation is, the granules of melanin and other colorful substances that gives you a brown animal, an orange animal, a black animal, or whatever. And often, just by looking at that, you have no trouble at all identifying what you have found. If you, for instance, see the one in the bottom, the number 34, the interrupted one, is typically what you would see in the hair of a human, or indeed a primate. Some of the Oran-Pendek hairs that Richard have brought back from Indonesia have these interrupted medulla pigmentation, which means it's, at the very least, it's a primate of some kind. Whereas in other animal species, you find something, number 33, just to the left of it. It looks like an old dry stone wall. That's very typical for animals like deer, goats, sheep, and all these vegetarians. There's various other possibilities, and by combining that with the surface structure of the hair, you can get quite a long way. Another one that's very important, strangely enough, is the margin between the central part of the hair, the colored part, and the outside, the outer layer of the hair. It can either be straight, or it can be fringed like in the middle, or it can be scalloped like the right one. And this is something you should pay particularly attention to if you are a big cat researcher, because all cat hairs, each and every one of them, and I have actually studied the hairs of every single known species of cat. They all have this fringed margin between the medulla and the outside of the hair. The actual, well, in some species, the margin looks like a hacksaw blade or a blunt saw blade. In other cases, they look like the end of a comb, a fine-toothed comb. So if you find a hair with these fringed margins, then you found a cat hair, and you're well on your way to an interesting discovery. And that's perhaps the strangest of them all, that you can also try to make a cross-section of a hair. I will guarantee that most of you would have thought that a hair is round, or perhaps oval in shape. For most of us Western Europeans, our hairs are round in a cross-section. If you've got curly hair, it's because your hair has an oval cross-section that makes it wave. If you're African, you have very curly hair. That's because every single hair is almost flat. But there's lots and lots of other variations, like you can see here. For instance, at the bottom, there is the, to the bottom right, there is the H-shaped hair. A strange, almost square type of hair with long furrows in them, which is actually the kind of hair you would find on a common shrew. But as you can see, there's all kinds of other variations, strangely, I don't know, cookie-shaped and whatever hairs. In some animals, sloth hair, for instance, also has these long furrows, but in the sloths there's actually algae growing in these furrows. I've never found algae in any other animal like that. You do find algae in hair samples when you find them out in the nature, but that's because they have been lying out there in the wild for far too long, and things had started growing on them. Yeah. And this is just to show you some of the things that you can find and what it looks like when you put the hair under a microscope, and also what enables you to rule out various groups of animals. These are all rodent hairs, and seeing the fact that half of all the, well, no, not really, something like what, 2 2/3 of all known mammal species are rodents. You can rule out quite a lot. They all have this strange medulla structure looking like a stack of cookies or cheeses on a shelf or whatever you like to call it. So if you find one of these, it's a rodent, so you can probably forget all about it. You can also find some very, very strange hairs like these, looking like plant stems, almost zigzagging back and forth. These are bat hairs. Every species of bat have these strange zigzag-like hairs, which sometimes give rise to rather frightening situations. I shall elaborate on that story. One day I had a phone call at home, and I was told by a friend of mine that she had been to the doctor with her daughter, and they had found out that she was allergic to animal hairs. And she was a bit worried about that because they had no pets of any kind, and they were living on the 10th floor of a high-rise building in central Copenhagen. But now she had actually seen some hairs on the back of one of their sofas. So would I come and take a look at it, because they were very short, these hairs, and she was wondering whether they could have mice or anything up there on the 10th floor. So I went out there and collected some of these hairs, and it turned out to be bat hairs. And I called her back and told her that there had been a bat in their apartment. About 15 minutes later, my doorbell rang, and outside was this friend of mine with her daughter, and telling me that she absolutely refused to get into her apartment again until I had been out there and removed that bat. And I said something along the lines of, "You must be out of your mind." And, well, we have known each other for some 25 years, which I think is the only reason she didn't knock me down on the spot. So I went out there, finally, and went through her apartment. Never found any traces of the bat, apart from the hair. I don't know how it got in there at all, but there had been bats in there, and apparently when I collected some of the hairs and gave them to the doctor, and he tested her daughter with these hairs, and it turned out that she was allergic to bat hair. Here's another type of hair, looking very much like this dry stone wall thing that I told you about. In this country, it's quite interesting and quite valuable to know what they look like, because that's how every single species of deer, how their hair look. So if you find one of these, it's a deer. And if you don't find, or if it isn't a deer, it's another big vegetarian animal, plant eater, a sheep or a goat perhaps. Here's another strange type of animal hair. You can see the entire hair is almost filled with these pigmentation granules. Most of the hair is one big medulla. And that's also a good one to know if you find a hair with any color in it, because that's a rabbit hair. And in this country, I guarantee you will find rabbit hair in almost any kind of environment, in scats from all kinds of animals, and well, just about anywhere you can think of it. Another good one to know, you can see this hair is another way of identifying it. In some hairs, the medulla takes off all of the hair, just like the rabbit I showed you before. In other animals, it only takes up the central third or something like that. This one is a strange mixture of the dry stone wall pattern I showed you before on the deer hair, and it has very fair, almost straight edges with a little scalloping here and there, and something that at the center on the underside of the center, you can see something looking like a big crack. That's very typical for a fox. And all these pictures are, of course, in black and white. But when you are studying the actual hair, you can always go by the color, of course. And here's a couple of rather important and interesting hairs, probably the most important ones for a cryptozoological researcher to know. And this one, you see about a third of the hair has color in it. The outer layers have this strange striation, almost looking like tree bark. And the margins between the colored part and the lighter part is almost just like a wavy line. It can also be completely straight. That's the hair of a dog, and almost every single breed of dog has hair looking like this. And I think at least 2/3 of the hairs I've been looking at this weekend have been dog hairs, so that's an important one to know. And perhaps at this stage I should also tell you a little bit about how do you do this. Do you need a lot of fancy equipment? Do you have to be able to split the hairs or stuff like that? Or do you need a 20,000 pound microscope like the one we have up here? But in actual fact you don't. You need a microscope, and you need a microscope with a magnification of, say, at least 200 times, preferably 400, but you can do well with a 200 times magnification. And apart from that, you don't really need anything. You can easily take one hair and study it. You don't have to split it or encase it in things and cut it in fine slices and stuff like that. You can just get onto it and study it. So it is within the grasp of anyone to try their hand on this. It is, as I've said, the differences are fairly small. It takes a little bit of practice, but you can usually do it fairly quickly. And then we have another very important one. The bottom one here, if you look at the margins here, you see this kind of saw-like fringed edge. That is a cat hair as well. So if you find one of these, then you should sit up and start to take notice, because that just might be something interesting. And when you have, when you've narrowed down the group of animals that you are actually looking at, cats, dogs, bats, whatever, then you have to, of course, you have to have a collection of hair samples that you know are leopard, lions, cows, cats, whatever. So then you have to compare with known samples, because it can be, if the hairs that you've found have started to degrade, it can be a little bit difficult to ascertain the exact species. But if you can compare it to leopard hairs or of a known source, or to lion hairs, or to whatever, I haven't actually told John McGowan this, but when I found these leopard hairs up in the forest, and I had analyzed them, and I was 95% certain that they were leopard hairs, I went down into the merchandise areas and nicked a couple of hairs from his leopard skin for a final comparison, just to make doubly sure. And I'm quite sure you can't see any bald spots on his skin, but I've confessed now, John, sorry. So you have to have hundreds and hundreds of little envelopes or boxes with all kinds of hair samples, and you also have to have a rather patient and understanding wife, as well as children who will keep their hands out of Dad's hair collection. And of course you have to be ready for strange calls from your children's teacher when they call and say, "Your son told in class last night that you have a large collection of hairs." I told him not to lie to his friends or to me. I would just like to tell you that has it been a problem for your son telling stories like that for a long time? And I said, dear Mrs. Hansen, he is telling the truth. I do have a collection of hair samples. And she was kind of angry and saying, you don't have to defend your son by telling lies as well. So next day I gave him 15 little plastic bags with hairs to bring to school to show the others. And about a week later I got a call from the principal at the school telling me that this teacher had suggested that the psychologist at the school should perhaps keep an extra eye on my son, because he was apparently from a home where things were a bit strange. So you have to be careful with what you do when you study stuff like this. But if you do that, if you practice a little with these hairs, you can actually identify all kinds of stuff. As you've seen this weekend, we've actually found leopard hairs here in the forest just a few miles from here, and actually have proof that there is leopards, or at least has been one leopard here within the last very few weeks. And we have enough hair should anybody else want to have a look at them and try to confirm my results, but I'm absolutely 100% certain that this is in fact leopard hairs. But you can also be called upon to analyze hairs from all other kinds of places and pretend that you are working for the police department, as when a friend comes with a sweater saying I've actually been contacted by a friend of mine who was certain that his wife was cheating on him, and brought me her sweater, where she has been out and asked me to look for any hairs that wasn't hers. And I told him that I am a zoologist. I would not want to interfere in his marriage in any way. They did actually get the divorce about 6 months later, but I had nothing to do with that, I guarantee. No, but the fact is that it is worth keeping a lookout for hairs of all kinds, and it is worth cultivating a habit of when you're out anywhere. If you find, if you go through barbed wire, through fences, through posts, through gates, all these kinds of things where an animal, a big animal would have had trouble getting through, and would have lost a few hairs brushing against a fence post or brushing against some brambles or anything. Take a look and see if there's any hairs sitting there, and if there are, take them home and try to identify them. If you can't identify them, send them to me, I'll identify them. Because you never know, there might be something out there, and if you keep your eyes about, keep your wits and eyes above you, you never know what lurks. And in this picture, you haven't got a hope in hell of seeing what's actually there. I realize that now. But, well, you can try and see if you can find the animal lurking in there. I did go back when it had disappeared and picked out some hair samples, just to be doubly sure that I actually identified it correctly. I haven't got a laser pointer, so I can't point to the actual creature, but can anybody see it?
Is it a car?
No, it's actually almost in the exact center of the picture. There's this very big dark, it's a tree trunk, and there's a light branch going across it. Just at the underside of the tree trunk, to the right of this light branch, is a round, reddish-brown blob. That's actually a mouse. And when the mouse had gone, I went over there and tried to find some hairs. I actually found a few, and it was, lo and behold, they were the hairs of a mouse. So I can only urge you, every one of you, to keep your eyes about, look where you're going when you're out in the wild and collect as many samples as you possibly can. Most of these will be of well-known animals, but every now and then you find one that is interesting, strange, out of the ordinary, and lo and behold, you end up here in the Weird Weekend talking about leopards and all kinds of stuff. And then, of course, when you have analyzed all your 257 hair samples, and it turns out that 250 of them are of, in fact, ordinary animals, then you're suddenly left with 7 that might be of interest, and then it might be worth to try to have DNA analysis done on them. And it's going to be considerably cheaper to have 7 analyses done instead of 257. So try this, and it can be quite fun and quite interesting. When you are going to do a DNA analysis on a hair sample, you need the biggest hair sample you can possibly get, as many hairs as possible, or you need hairs with the bulb, the root, as well, because that contains more DNA than the rest of the hair. And in many cases, you actually won't get any definitive result, because a hair that's been sitting out in the wild for perhaps weeks starts to degrade just like any other thing. Microbes, algae, fungi, all kinds of stuff starts to grow on them and starts to degrade the DNA. So you might not be able to get any definitive result, just like I haven't been able to get any definitive results on your oropendric hairs yet, because they were in a rather sorry state. They had been out in the hot and humid tropical jungle and probably started to degrade in the instant the animal dropped the hairs, and they started to degrade. So the fresher the better, the more the better, and then perhaps you can find something, or you can prove that wherever you come from and wherever there are stories of big cats, that there actually are big cats out there. Right, this is about as much as I'm going to say, because to go in any more details in this would probably bore half of you to death, and the rest would fall asleep. So I'd rather hand it over to you. Does anybody want to ask any questions anymore? Do you want me to elaborate on any of this? Or if you want to say that this is the most boring lecture I've ever heard, that'll be fine with me as well. No, I can't see any hands anywhere because of this spotlight. But you handle it, Greg. Yeah. Don't mind the cameraman.
With the sample you showed yesterday of the leopard hair,
Yeah.
is this on?
Yeah.
How can you tell between a domestic cat and a leopard by with the comparison? What's the difference?
There's various ways of identifying it, but one thing was that it has these fuzzy edges between the medulla and the central part and the outer part of the hair. That makes it a cat.
Right.
And then you have the width of the colored part compared to the outer part. Most big cats have a fairly narrow colored part and a very distinct outer cortex layer, if you like, whereas domestic cats, most of them have the hair completely filled with color. Domestic cat hairs are often very soft, and when you pick them up, they're kind of like flappy and wavy like that, whereas big cat hairs are, even the thinnest of big cat hairs are rather stiff as well. And there can be a slight difference. Big cat hairs can be a little bit thicker. You can measure them as well, but they can. It depends on where on the body find the actual hairs from. Back hairs are longer and stiffer than hairs on any other part. Facial hairs are very short, but there's various ways of distinguishing, and you can look at how these protein scales on the surface, how they are placed and how big they are, how rough they are. I've seen several samples of tiger hairs where these protein scales are so thick and rough that it looks like, it looks like you're looking at a miniature tree trunk.
Okay, thank you.
And Greg, there's someone on the back there. You can take that with you. Oh,
I was wondering whether your microscope needed top light or whether you could get by with light from underneath.
no, you can easily get by with light from underneath, unless every now and then you find hairs that are so black that you can hardly see anything. But then there's various things you can do. You can actually, with a bit of care, if you take a glass plate and smear a little nail varnish on it and put the hair on that, you can scrape it a little bit so it gets a bit thinner, and then usually the light can shine through and you can see what you need to see. But the only thing that's virtually impossible, that's guard hairs, because if they're colored, they are so thick and so full of color you can't see a bloody thing.
So the dog's guard hairs would be too dense, and the inner hairs that keep the dog warm would be the ones to go for.
Yeah. You can't, with guard hairs you have to, it's more a matter of measuring how thick they are, and how long they are and stuff like that. It's very difficult to actually see any kind of structure in those.
Can you make precautions to preserve the hairs? Making slides makes that make sense, but the hairs themselves, it's just a matter of keeping them away from the weather.
And yeah, basically, yeah. As long as you keep them dry and away from light, you have to keep them in somewhere dark, because if you leave them out in the sun, everybody knows how after a long hot summer your hair tends to get pale, and it's exactly the same with loose hairs. The color starts to degrade and fall apart, so you keep them dry and keep them dark, and they can last for years and years and years.
And how do you go about making a cross-section of a hair?
If you have to make a cross-section of a hair, then you need more, what should we call it, scientific equipment. Then you have to encase the hair in some kind of resin or plastic, and you have to make slices of this. And for that you need a bit more scientific equipment, special cutting machines that can actually make the slices so thin that the lights can shine through them.
My microscope, the geology one, it has everything to check for polarization. Do you get polarization effects?
Yes, especially for very light hairs, white or very, very light hairs. It can be possibly help you to get out more structure in the hairs. But you can easily make do with just an ordinary microscope with light coming to shine through the hairs from beneath. Greg, up the back there, Carl.
I know you said that if you can keep the hair dry and out of the light, you can preserve it.
Can we have a bit louder, please? So I can't hear. Try again.
I know that, yes. I know that you said that if you keep them dry and out of the light, it will keep for a long time. But most biological tissues have a preservative, a fixative that you can put in them to preserve them. Are there any sort of liquid fixatives or preservatives that you know that you could use for hair if you wish to make a slide?
No, I would just save them in a dry, dark place, because most of hairs are made of the same material as our nails and the claws of animals and so on, and that will keep for, well, almost forever. And if you want to make a slice, you can just pick out your hair off this whatever little envelope or box colours you got it in, and then put it in, encase it in plastic or whatever you like, even 10, 15 years later. The problem is that if you put it in some kind of preservation fluid, formaldehyde, alcohol or something like that, there's a risk that the colours will bleach out. And that can make it more difficult to actually identify the hair. In some cases, it's a good idea if you get a hair which is extremely dark, you can try to bleach it a little bit to wash out some of the color so you can see the structure, but you have to do that very carefully because you can also, if you leave it in there too long, wash out all the colors and you won't be able to see anything. But I would still keep them dark and keep them dry, and then you can hand them over to a lab in 15 or 20 years time and they can do it whatever it is, and there would be no problem at all.
Thank you.
Hi Lars.
Hi.
Very general question for you. What is your most interesting / exciting discovery that you've made?
You mean apart from the leopards yesterday?
Apart from that. Yeah, that might be the top one for you.
Apart from that little trifling incident, I did one. I did one. They find, and that was about the strangest things that I have ever experienced. I was in a department store in Copenhagen, and I was standing at a counter, and this girl has gone for something that I wanted to buy, and she didn't exactly have it there. And I was, like you do when you wait around and you look up and you look down, and I looked down and noticed a hair lying on the floor. So I picked it up and I thought, that's strange, because it was very, very thick and very stiff. And I just put it in my pocket and went home with it. And cut off a piece because it was rather long. Cut off a piece and put it in the microscope, and I thought, what the? And it took me a while to work that one out. It was, in fact, a rhinoceros hair. I haven't got the faintest idea how a rhinoceros hair ended up on the floor in a department store in Copenhagen, but there it was. I'm certain it's no big scientific discovery, but it is really, really strange.
Rhino horn is compressed hair. Could somebody have got some illegal rhino-based rhino horn artifact or something? Did it come off?
Well, I don't actually think so, because rhino, I've studied the structure of rhino hair as well, partly because they are compressed hairs, but they are not, though they are hairs, they don't have the normal structure of hair from the body. So I don't
sort of welded together.
Yeah, and they don't have any of those protein skates on the surface. They don't have them, and there's not as much color in them as there is in the body hair. So I don't think it could have been anything illegal in any way. Maybe someone had been a tourist in Africa and bought a bracelet made of rhino or elephant hair and had dropped part of it or something like that. I haven't got the faintest idea.
Sounds like the only possibility.
Yeah.
Anything else other than teleported rhinos?
Oh yeah, well, sometimes there is a lot of heavy stomping and grunting in the streets of Copenhagen, but
Max.
How would you identify pangolin?
pangolin hairs, they look
Yeah, because the Asian species have a number of hairs sort of underneath the scale.
Yeah, they look like railway tracks.
Under,
Yeah,
so fairly easy.
yeah.
Max, forgive me. Why did you ask that question? That's the most obscure question I can imagine anyone ask.
Because no one else would.
Fair enough.
No one else likes pangolin.
You can see why I've taken this boy under my wing. He is an original thinker. Anybody else? Oh, blimey. So who we got? Stuart, back.
Mine's just a quick question about the Wolseley leopard that we've got in the be a superstar before too long. You mentioned about proteins deteriorating from the hair sample. Over what period will that deteriorate as such? I.e., how long was it there, and then I said to go walking tomorrow night afterwards?
It's, well, those protein scales are very thin, so they disappear quite rapidly. The rest of the hair is extremely durable, so that can last for years and years and years. And it also depends on the weather, the temperature, the humidity, the moisture, I would guess, but please don't hold me responsible for it, that the hairs has been out there for at least a week, possibly 2, or something like that. It's not an animal, this animal hasn't been through there within the last couple of days. A week, 10 days, perhaps 2 weeks, something like that.
Richard. Richard and then Helen.
Hi, what's the diagnostic features of marsupial hair?
Marsupial hairs have a very strange structure. Unfortunately, I couldn't find any pictures of marsupial hairs to bring before I got here, but the pigmentation, the pigment granule inside the hairs are not these detailed structures. It's more like irregular lumps of color just lying about, like someone had taken an ordinary mammalian hair and shook it about until all these pigment granules are just broken apart and laying all over the floor. It looks more messy and yeah, well, just more random somehow. And then there's various other differences. Of course, you can separate kangaroos from koalas and wombats and so on. But the main feature is the fact that these colored granules are just, they have various sizes. So some of these I showed you looks like a stack of biscuits, but in a marsupial hair you will find there's a big blob here and 2 tiny ones and a medium-sized one and then a big one and then a part of the hair where there's no color and then a few other bits and blobs along the way.
Helen, yes.
The protein scale, did they help you differentiate between a live subject and for a coat or something?
Not necessarily. You can do chemical analysis on hairs to see whether they've been treated in any way, but that's rather difficult and complicated. The easiest thing is that when you sometimes you find hairs with a very strange uniform coating on the outside, and in every single case it's turned out to be woman's hair with hair color or stuff like that on them. But the actual, if you mean, it could be hairs from an old skin or from an old fur coat or something like that. It takes a fair bit of chemical analysis to be able to ascertain that with any kind of certainty.
Any more questions? Okay, we have, I can't see who, yes.
There's one down by the front.
What about the hair from hybrids? I mean, have you managed to collect samples from those, and how do they differ from the parent animals?
No, I've seen in one case, I've seen one tuft of hair from a cross between a lion and a tiger. That looked like lion hairs, very rough lion hairs. Presumably the hybrid has the rough surface of the hairs from the tiger, and for the rest it looks like a lion hair. I've seen hairs, the only other hybrid I've studied is hairs from mules, and they look very much like donkey hair, but are a bit more stiff and rough, more like horsehair. So they have the general outline and structure of a horse, but the internal structure is more like a donkey. But they can be definitely tricky to determine those.
I tell you what, something has just come to mind. Is there a parallel between the diagnostic capability, not capability, is the wrong word, the way that you can diagnose microscope, if you look at them under a microscope, do the feathers of the families of birds differ?
You can identify the various families of birds. Whether you're actually able to identify the exact species of a bird from just a tiny fragment of a feather, I don't know. But you can. The structure, the general structure of the feather is, there is a variation between the various families, whether it be a duck or a passerine or a bird of prey or an owl or something like that. You can do that just from a little bit of feather. But usually when you identify feathers, you have the whole one, so you get...
Yeah, exactly. I'm just wondering if you just have one strand of it.
Yeah, you can get to the family, but whether you can actually get to the actual species, I don't know. I've never tried it more than to family level.
Richard?
I'm aware from eating chickens and turkeys and so forth that birds have hairs too. I know that the monitoring should have interesting hairs, although I'm not quite sure how you manage with echidnas. I'm not sure if echidna and, say, hedgehog have hairs as well as their spines.
Yeah, they do have hairs. I've never had a chance to study their hairs, so I don't know what they look like, but I would imagine something rather primitive and random, just like the marsupial hairs, but I've never had a chance to study any of these. And for what looks like hairs on chicken and so on, is in fact the early stages of down, very, very primitive feather-like structures. They're more feathers than actual hairs.
There's a fashion for keeping cats which are, well, they're a hybrid of domestic cats and an exotic species of little cats, where the original first generation hybrid has then been crossed to domestic cats in several generations. These things, generally speaking, they act like domestic cats but a little wilder. They do have a significant portion of the original exotic cat's genome in them, which you see from various digestive maladies and things like that. But do the hairs on these things differ significantly from domestic cats?
I've only seen the hairs of one of these, and that particular individual had hairs that looked exactly like its wild ancestor. It was a cross between a domestic cat and a South American leopard cat, and that one looked exactly like a leopard cat when you just looked at it. It was a bit more gentle and had a nice and friendly disposition. But it looks exactly like a leopard cat, and the hairs looked exactly like a leopard cat, because I went out to the museum and took some hairs from one of their leopard cat skins to make a comparison, and I couldn't see any difference at all.
Anyone else?
Oh, Lars, I just wanted to know how big...
Wait for the mic.
Okay, sorry. I just wanted to know how big your collection is, because it sounds like it must be enormous.
You don't want to know. No, it takes up several, file boxes. There's heaps and heaps and heaps of little plastic bags in every one of them. It's not that big when you just see those 4 boxes on my shelves, but if you open them, there's several hundred of these little plastic bags in every one of them. So, but right now, actually, they're deep in an enormous pile of other big boxes, because a couple of months ago, 6 months ago, I had to move out of my flat because of renovations, and I've only just moved back fairly short time before this, so they're in box number 43 in the back of 6 row of boxes, I think. So they're not immediately available, but they are, I think, 4 filing boxes like this filled with, I don't know, 800, 1200 little bags, plastic bags.
And I guess secondly, would you like any new hairs for your collection?
Yeah, well, as long as they fit in those 4 boxes, I think I'm allowed to bring in more, but if I start enlarging the collection, my wife might be, what do you think you are doing?
Anyone that wants to do this will need access to literature. What would you recommend in terms of how people, which literature do you think they should use and how should they get it?
The literature that they should use is, well, they should start with a book by a Dutch zoologist called Hans Tiebrink called Hares of West European Mammals. That book has a detailed description of how to do the analysis and all the various variations in the medulla, the pattern of the surface of the hairs, and you can buy it on Amazon. And when I bought mine, it was some, I think, 28, 29 pounds or something like that. So should anybody like to do anything like this crazy stuff, they should buy this book. I got a copy of it with me upstairs. I can go up and bring it down if anybody wants to have a look at it. But that would be the book I would recommend.
Anyone else? Well, Lars has been my friend for I think 16 or 17 years. We've known each other a hell of a long time, and it's always a pleasure to have him here. I think the last time you came here was 2 years ago?
No, I was here last year as well.
You were here last year, then the year before that you weren't. The year before that you were here at Karina's and my wedding.
Yes.
Last time. And